Hello everyone, myself Dr. M. Sumitra from School of Pharmaceutical Sciences, VISTAS. Now I am going to discuss about pH. pH is an important chemical term. It is routinely used in our pharmaceutical field. So if you are taking any substance, by applying the pH determination, we can know whether acidic or basic or neutral substance. So in our pharmaceutical area, we are used number of methods to determine the pH. So we are now learn one by one. In my presentation, I have discussed about what do you mean by pH and how to determine the pH of the solution and what are the application. So first of all, I say about what do you mean by pH. pH stands for potential of hydrogen or power of hydrogen. It is a numerical scale used to measure whether the substance either become acidic or basic or neutral solution. So if you are taking the pH scale, it will be covered between the 0 to 14. So we are represented only the numerical value. So if you are lesser than 7 mean the solution become acidic. So the basic example for acidic solution is lemon juice, vinegar, or in our lab we are routinely using the hydrochloric acid, sulfuric acid, nitric acid. These are all the acidic solution example. So these are all will be example for stronger acids. If you are taking the glacial acetic acid, phosphoric acid, these are all the weaker acidic acid. So these are all will be, if you are measured by using the pH scale, it will be become the lesser than 7. So if we are getting the exactly 7 mean, the solution become neutral. So the best example for neutral solution is our pure water. So the pH of our water is 7. This is the neutral solution example. And the next classification is basic solution. So if you are taking the pH more than 7, that will be called as a basic solution or alkaline solution. So we are routinely using soap, baking soda. These are all the best example for basic. In our lab, we are using the sodium hydroxide or potassium hydroxide. These are all will be the ammonia. These are all will be the example for basic solution. So if you are taking the formula for pH mean, pH will be represented as negative logarithm of H plus ion. Here the H plus will be represented as concentration of hydrogen ions in a molecules. So now we can learn how to determine the pH. So if you are taking the determination, we are doing the 3 types of method. So if your first one will be the manual method. So in that method we are using the pH paper. So in that pH paper, just the pH paper will be dip into the your unknown solution. It changes the color. So according to the changes of color, it represent the pH. Another method will be indicator solution. We are put the drops of the indicator solution, it changes the color. So according to the changes of the color, it represents the pH of the unknown solution. So these two will be coming under the manual method. By using pH paper as well as indicator solution, it is coming under the manual method. So this is the approximate method. Just we can get the idea about our unknown solution. Either this solution will be acidic or basic or neutral. So another third method will be by using the pH meter. This is the instrumental method. So we are getting the exactly what is the pH of our unknown solution. So this is the pH paper. Actually the pH paper mean is a normal our cellulose paper, but it impregnated with the our indicator dyes. So alkaline dyes will be there. So it will be coated with that paper. So just if you are dip into the our unknown solution, it changes the different color. So along with this paper, they are giving the color shades. So it represent the pH of the solutions. So if you are coming the red color mean it will be the acidic, slightly yellow color it become the weakly acidic, then blue color, dark blue color it become the alkaline solution. So this will be the coming under manual method. This is the another method. By using indicator solution, we can determine the pH of unknown solution. So number of indicator solutions are available. Either phenolphthalein, methyl orange, methyl red. If you are taking the indicator, it is a weakly organic acid or weakly organic base. So it will be react with your unknown solution. Then the ionic changes will be occur. So according to the ionic changes, it shows the color change. So if you are taking the indicator, it will be available different form. Either it will be the single indicator or mixed indicator or universal indicator or screen indicator, like that numerous number of types are there. So and even it will be according to nature, they are classified acidic indicator, basic indicator or neutral indicator. So the function of indicator solution is just you put the drop of the solution in unknown solvent, it shows the color change. According to the color change, we can easily identify what is the nature of solvent. So if you are taking phenolphthalein, it is a basic indicator. If you are add to the acid solution, it will be the colorless. But same phenolphthalein, if you are adding to the basic solution, it becomes the pink color. So according to the color changes, we can easily identify the your pH of the unknown solution. So this is the best example. Bromothymol blue is a universal indicator. So the color changes will be become from the acidic to base. So if you are taking the acidic solution, the bromothymol become the colorless. Then slightly yellow color if you are become the weakly acidic. If you are near to the 6.4 pH, it become the lemon yellow color. Then slightly it change to the blue color, that will be become the 7. If you are more than 7, the color of blue color intensity will be gradually increases and become dark one. So we can easily identify the solution become basic. Now we can learn about instrumental method for pH determination. So we are using the pH meter. pH meter mean is an electronic device that measures the acidity or alkalinity of a unknown solution by detecting the voltage difference between two electrodes. So if you are taking the pH meter, it contains the two electrodes. One is indicator electrode, another one is reference electrode. So generally in our instrument, the indicator electrode will be made up of glass electrode and the reference electrode will be made up of hydrogen electrode, and even the temperature sensor also there. We can adjust the room temperature, and also even it will be changes the temperature to the either increase or decrease, and another part will be the display, pH meter display. So if you are taking the instrument, the indicator electrode will be, if we are using the glass electrode, it will be made up of the glass membrane. In that electrode, the glass membrane will be there. So just if you are touch the solution, it shows the pH changes. So this will be the act according to our unknown solution. But if you are taking the reference electrode, it shows the standard pH. So it will not alter according to the unknown solution. It will be act as a standard reference. So if you are coming to the principle, actually the ions are exchanged from the sample solution to the inner solution of your glass electrode via the glass membrane. Via glass membrane, your unknown solutions are enter into the glass electrode. So the pH changes will be happen. So according to the changes, it shows the pH of your unknown solution. Even if you are taking the pH meter, pH probe will be there. So it just touch the solution, it shows the signals. What is the pH of our unknown solution? So next we move on to the what are the importance of pH in pharmacy. So if you are taking the pH, it will be having the basic in our analysis. So it shows the numerous activity in our analysis like determination of drug solubility, drug stability and absorption and bioavailability of our substance during drug formulation, even preparation of buffer system. So if you are taking the solubility, the some of the drugs solubility depend upon the pH of the medium. Either some substance will be dissolved in a weakly acidic medium or even some neutral medium or in basic medium. Example, if you are taking the aspirin, is more soluble in our alkaline pH. Like if you are taking the drug stability, some of the drugs it will be stable in a particular pH range. If more than that or lesser than that, it will be not stable. Either it may be goes through the degradation or hydrolysis or oxidation, anything will be happen. Example, if you are taking the penicillin is unstable in acidic solution. The next important thing will be the absorption and bioavailability. In generally, some of the drugs will be stable in our GIT, some of them will be stomach. So it will be the live example for the pH is how much importance in our protein life, even in our pharmaceutical field. Some drugs will be absorbed in acidic, some will be basic. Another important thing will be drug formulation. During the formulation of drugs, the pH affects mainly excipients, selection of preservatives, flavoring agents in dosage forms. And another thing is buffer system is very important in our analysis. The selection of buffer will be plays an important role in our chromatographic, even spectroscopical methods. So by determining the pH, we can know the buffer pH solution. So according to that only we can select the solvents. So these are all the reference for my presentation preparation. Thank you.